Sure, thanks, Lauren. This morning, I'm really excited to introduce Dr. Pejman Rohani. Dr. Rohani is the Athletic Association Professor in Ecology and Infectious Diseases and is jointly appointed at the Odom School of Ecology and the Department of Infectious Diseases in the College of Veterinary Medicine at the University of Georgia. In 2019, he was elected as a fellow in the American Association for the Advancement of Science for contributions to the field, including infectious disease dynamics, epidemiological theory and modeling, and model-based inquiry. And really, he's just been an incredible leader in the field, particularly with contributions to mathematical modeling of infectious diseases as a whole, and his fundamental research to learn about influenza and pertussis transmission dynamics, as well as a number of other infectious diseases. Many of you may also recognize his name and be familiar with his book, which I think is a must-read on epidemiological modeling of infectious diseases. And he also released a book that is really a tour de force on pertussis that he edited alongside Sam Scarpino. He's also, not just a leader in research, but also has been, really an amazing teacher and mentor in the field. And I met Pej the first year of my PhD and was lucky enough to get my first introduction to mathematical modeling from him and John Drake at SISMID. And I really highly recommend his materials from the class for anyone wishing to learn more about modeling. And with that, I'm looking forward to learning more from him about COVID-19 this morning.
You're muted.
Thank you for the kind introduction, Spencer. And I was told that I can give a presentation that is speculative in nature and ongoing work. So that's exactly what I'm going to do. And I'm going to beg for your indulgence in what's about to ensue. So my co-conspirators in this project are one of your alum, Shweta Bansal, primarily Toby Brett, who is a postdoc in my lab. We're focusing on COVID-19 in the state of Washington, trying to fit metapopulation models and computational statistics methods in order to have an understanding of the spatial spread of the virus in this population. So just a little bit of introduction in terms of what happened in this population. January 21, last year, 2020, CDC confirmed that a 35-year-old man from Snohomish County, I'll show you a map in a moment, had been visiting his family in Wuhan, China, returned back from China without any symptoms. He arrived on January 15th, and then on January 19th, he reported to an urgent care clinic, and he was released from hospital on February 3rd after 2 weeks of treatment, including remdesivir, and then he went on to into isolation at home. It's quite likely that there were previous cases in Washington State from sequence analysis dating back as far back as 2019. So just for those of us who are not particularly familiar with the terrain, this is Washington State, a bunch of counties. We have plotted here the population density per square kilometer on this map, and I'm going to be talking about a number of focal counties. So the state population is about 7.6 million. King County is where metropolitan Seattle is based. It's about 2.2 million population size. Snohomish is just above it. So we're talking about this kind of corridor on the west of the mountains. Snohomish has got about 820,000 population. Pierce County just below is about 904,000 population. And, to the east, we have Spokane County with about 500,000 population. So that's just to orient you given some of the data I'm about to show. The time series for the entire state looks like this. So we're looking at data coming up to almost up to date. In blue, I'm showing you the confirmed cases across all ages, and in orange, I'm going to show you the hospitalizations in individuals younger than 60 with the axis on the right-hand side. So available to us are daily reports of confirmed COVID-19 cases and hospitalizations from the Washington Department of Health. And in terms of the counties that we've looked at, King County has these very clear 3 waves. If we look at the blue line on this graph, with a couple of kind of modest size outbreaks in the spring and summer, and then an enormous outbreak from Thanksgiving onwards. The hospitalizations in this population were kind of tracked the the transmission activity, but the summer peak was smaller. In Pierce County, we get something very similar, but the hospitalizations were a little bit bigger in comparison in the summer. Snohomish kind of had a very, very slow takeoff initially, but then had a big outbreak in later on in the year after Thanksgiving. So I say we wanted to take a look at epidemiological connectivity in Washington state using mixed population models. And it's worth mentioning that the state issued social distancing guidelines on at the end of February. So in most of these time series, you can see, for example, that there's a downturn in these in this initial wave, which one assumes is attributable to the stay-at-home ordinances that were issued in the state. So one of the things that we did was to look at pairwise correlations using Spearman's row. And we can do a clustering analysis of this matrix of correlations. And there isn't really a good theoretically informed way of doing this, but we found that kind of the maximum distance in terms of this correlation coefficient that we're seeing is about 0.7-ish, 0.74. So we drew our threshold for this maximum, for this distance to be about half of that. I say somewhat arbitrary, but as we move that threshold, obviously we're going to get a different number of clusters. So I don't want to argue that somehow I would testify to Congress about this. Not that testifying to Congress is a big deal these days, but based on this somewhat arbitrary threshold, we end up with the following clustering of pandemic patterns in Washington state. So it looks a bit busy, but we can essentially point out that there are 3 clusters, the green, the blue, and the orange, which are really about counties that are really close to each other, they're contiguous counties. And yellow is kind of this high dense area around Seattle and that I think it's I-95 corridor. One weird one is that there is a Wakiakum County in the southwest of the state, which somehow clusters with this group. We also find there's a couple of counties which are very small and distant from each other that are the most similar in gray. And the pink cluster is essentially everything else. And these are counties in which the epidemic took a long time to get to. And typically, like the data that I showed you for Spokane, they kind of have a very small first wave, a very small second wave, and then an enormous outbreak in the third wave. So beyond this kind of a descriptive analysis, we wanted to do some model-based inference. And so I'm hoping that this is an equation-safe space. So I will spend a few minutes discussing the model. Our model is kind of inspired by the TSIR framework that Grenfell and colleagues have pioneered, but it's got some important differences. So our model is in discrete time. A time step is a week. So the number of susceptibles, we're ignoring demographics in this population, births and deaths in the fitting. So in a population I, in a county I, during week T, the number of susceptibles during week T is going to be however many susceptibles there were the previous week, minus those who got infected. So that's this C of TI variable. And so the number of infectious individuals this week are the ones who have been infected. And this C of TI is this random variables given by a Poisson distribution with the expectation that you have a force of infection, which is lambda of TI, multiplied by the number of susceptibles in that population. And then we have an environmental stochasticity parameter. So our model has demographic noise as represented by this Poisson process that's generating the number of new infections. But we also have this environmental stochasticity term that's, that is included in the model. So we now need to, this equation has a lot going on in it. So we need to unpack it. And in particular, the important thing for us is the force of infection. There should be a lambda of T comma I. So the force of infection is going to have two components. We're going to allow there to be, in the first term, we're going to allow external importation. This is external to the state, right? So the second part, the beta ij's, is to do with how much commuting there is among counties in Washington state. So that's kind of internal coupling. This is individuals flying into Washington state or driving into Washington state from some other area and importing the virus. So eta is this external importation, and we're going to estimate that from data. And this Ri of T parameter is the local transmissibility function. So it's at time 0 is going to be essentially our R naught in a population. And we're going to describe that by some basis with a spline. So what we could go in and put in information about the severity of social distancing measures implemented, try and estimate compliance and the effect of that on transmission and so on. But that's not really what we're interested in. So what we're going to do is we're going to have these flexible functions that are going to let the data tell us the effect on transmission whatever it was, the local ordinances that were put in place. So we're going to have a bunch of basis functions, b subscript mu, and they're going to be estimated for each county. And this A0 is going to be essentially our baseline reproduction number, our baseline R0 in our model. And so we assume that before social distancing, before the end of February, as I mentioned, all of these coefficients are 0, so we just have the baseline reproduction number. So they will look something like this. So before the 1st of March, we have a baseline transmission rate in each county, which we're going to estimate. And then after the rollout of these social distancing measures, we're going to estimate what this flexible spline function is going to be given by the data. So this is looking at the temporal component of the model. We also have the thing that we're kind of interested in, which are the spatial coupling. And our beta ij is going to give us the per capita transmission rate from individuals resident in county I to individuals resident in county J. And it's a summary of the effects of social distancing and mobility patterns. And so it's going to be given by this slightly weird looking expression. So we have this, I called it Ri in the previous slide, but now we're going to give it subscript Q because we're going to be summing over all the counties Q. So the idea is you have individuals from population J going to population Q, and individuals from population I going to population Q, and this is where they're meeting, right? So that could actually be population I, it could be population J, but we're looking at movement from different populations and how much transmission is going on within that population. So that's going to be summed across all the counties, is going to be the transmission rate from population I, county I, to population J. This K matrix, the entries of that are going to tell us about the probability that an individual who resides in I commutes to Q, sorry. And we get this from the 2010 census data, where they actually have commuting flows between counties. So we end up with a slightly gnarly looking matrix, which has, if I and J are not the same county, then you end up with this expression. And if they're the same county, you end up with that expression. And so this Nij is, again, the number of commuters to population J that reside in population I. And this NQ tilde is really the individuals from population, from a population I in this case. So this is the total population size, this the fraction of them that are currently in Q, commute to Q, summed over all I. So this is like all the expat population that are currently in population Q. So all of that is to say, that's just kind of the bookkeeping of people moving around. And we have these data. This is the scaled Nij matrix that I mentioned. And so, for example, not surprisingly, this matrix is very strongly diagonal. So this is the proportion of contacts from individuals who live in that county with individuals in that county. So, for example, most of that ends up being like in King County 9.97 end up being within individuals in that county. So we can parameterize a bunch of the spatial coupling matrix from these covariates that we have from the census data. And there are other things that we need to calculate. For example, this epsilon parameter I'll show you shortly, that we need to estimate from the data along with the transmission function. So, and then we have to figure out, now that we have a model, how do we turn our model into probability density so that we could confront it with the data and a likelihood-based inference. So one of the things that we found was that it was very difficult to get a handle on how reporting probability changed through time, right? Testing frequency changed through time, the diagnostics improved through time. And so in order to, rather than fit to actual case numbers, which are much more prone to changes in reporting procedures, we wanted to look at data that were, in some sense, the least variable in terms of their likely reporting. And we settled on the hospitalizations reported in individuals younger than 60 for that purpose. And one of the things that we found was that the age distribution of these data, which I'm not going to show you, really very rapidly converged to the stationary age distribution. So after just a few weeks, the age distribution in this category, in the hospitalized younger than 60, really didn't change all that much through time. So again, that's one reason to choose this rather than looking at actual cases. So we assume that the hospitalizations, that the data are given by an overdispersed normal distribution with this mean and the standard deviation. So the mean is given by a reporting probability multiplied by that C of Ti, which I showed a couple of slides ago. And the variance is given by, this is kind of a binomial if you just look at this first term here, but you have this overdispersion in the reporting process described by the model. So overall, our model is a Markov jump process, which we can't solve analytically. So we use sequential Monte Carlo particle filter to approximate the likelihood. So much of this was done in MIF, which is part of the POMP package developed by Aaron King and colleagues. So let me show you some fitted, the results of some fitting. So King County, again, this is where Seattle is, and we're looking at the number of hospitalizations per 100,000 in the orange line. And our fitted model gives us this time varying reproduction transmission. Well, it's kind of an effective reproduction number, which we call the local transmissibility. And so there are times when it drops below this threshold of 1, and in the build-up to the outbreaks, it exceeds 1. And so we've been able to, we haven't been able to fit a model that is comprised of all of the counties in Washington state. And that's because we've not been, that requires the introduction pattern to be introduction of the virus following, say, the initial introduction to be well described by the commuting data, and we've had a problem actually fitting the model to all the counties. So it becomes, it's in part a question of dimensionality, and in part it becomes a problem of the data in some of those really small, far away counties being too sparse in terms of the hospitalization data. There are more numbers in the cases, but hospitalization data end up being quite sparse. So the patterns are pretty consistent among most of the big states, or the big counties. These are all the ones in and around Seattle. Thurston, which is much further to the south, we find there's little transmission going on for much of the period, and Kitsap, which is the other side of King and Snohomish, again one of the smaller counties with about 270,000 people, has again this kind of these two little peaks later on in the data. So one of the first things that we want to do is to have a sense for how confident we are about the point estimates that we're getting, and how precise those estimates are. So what we're going to do is look at likelihood profiles. So this will look a bit weird to start with, but if you bear with me, I'm going to kind of build on this picture so that ultimately it will make sense. So in the x-axis, I'm going to change the baseline reproduction number. That was this parameter A0 in the splines that I talked about. On the y-axis, I have the probability that you have a case under 60 that's hospitalized and reported. And I've got two other parameters. I've got this, how many contacts you have in your commuting destination. So individuals are moving to this place where they're meeting other people. And I have how much external import of virus there is into the whole state, to the whole meta population. So I'm going to set this to 0. I'm going to set this to 0, but I'm going to vary this probability of hospitalization and this baseline reproduction number systematically across going to do a two-dimensional parameter sweep. I'm going to calculate the likelihood of my model using a particle filter, and I've got the likelihood shown here. So for this, if this is 0 and this is 0, across this two-dimensional space, I get this surface of likelihoods, and where the black dot is my highest likelihood for this parameter value. Now, keeping this eta parameter still at 0, I can then systematically change epsilon in increments of 0.1. And one of the things I notice is that as epsilon gets bigger, these likelihood surfaces are getting lighter, which means my log likelihoods are getting bigger. And so the model is explaining the data better. And then I can actually go through and do a full 4-dimensional likelihood profile, where I'm looking at these likelihood surfaces across changes in the external introduction, in terms of the commuting destination and hospitalization and the baseline reproduction number. And actually this yellow dot is my global maximum likelihood, and these black dots are the highest likelihood for each of those two-dimensional likelihood plots. The dotted red line corresponds to essentially the probability that case is reported under 60, matching, confirming with the data that we've seen from Washington State, right? So estimates that are very different from what's from the red line we're generally going to be slightly suspicious of. But in general, in all of these plots, the highest likelihood is in and around the value that is consistent with what's been estimated from other data. Now we've got this 4-dimensional likelihood, and we can collapse all of this information into single likelihood profiles in parameters that we care about. So we have a baseline reproduction number. So this is before the introduction of social distancing. We're getting essentially an R0 of about 2.6, which is consistent with other estimates at the start of the pandemic. The confidence intervals, so if you go down 2 log likelihoods, the confidence intervals are reasonably precise. We find the probability of having a case in an individual under 60 that hospitalized is just above 0.1%. Again, that's consistent with the red line, which is from independent data sources. The probability of contacts in the commuting destination, actually we find the MLE to be 1. I think we should probably, one of the things we need to do is do a finer sweep in this area to see if that really is the MLE or not. That slightly surprises me. And the external introduction rate, actually we find the MLE to be 0. So once the epidemic started, there is no signal in the data that individuals coming into Washington State with COVID impacted the epidemics that we see. So that's how I interpret that, this result. So these are our estimates and how confident we are of these estimates. And like, as usual, it doesn't take very long to show all this stuff, but it took a very long time to do 4-dimensional likelihood profiles. So it's kind of heroic work on the part of Toby, but I think this is very useful in knowing how well behaved is the estimation. Now we want to see, we simulated our model 1,000 times, and we want to see how it compares with the data. So the data, as you recall, is this gold line. And for each week, we've essentially produced a probability density of our 10,000 simulations. And the red line is the median of those 10,000 simulations. And what it shows you is the data are taking off more rapidly than the median model simulation. And
sorry, can you just remind us which data go into the simulation? What does the simulation know?
So the simulation starts with one individual in Snohomish County and everyone's susceptible, and then we just integrate the model forward. So it's not a one-step ahead forecast or anything like that. So it's just an initial value problem.
Okay, but there's mobility data, just no epidemiological data?
That's right. So we have the mobility data that parameterized our matrix,
Okay.
and that's in the fitted model.
Okay.
But it's not time varying.
It's not time varying. Okay.
No. So the only thing that's time varying are those basis functions that we have, the spline, that capture the effect of social distancing and reductions in transmission. What we can show, this is kind of, it may not be clearly visible on this slide, but if you look over here, for example, there is some light blue around this trajectory. There's some light blue around the data. So we can actually show that if you look at the top 5% in terms of likelihood of our model trajectories, they kind of track the data pretty well. It's just that the median of our model trajectories is underestimating the data quite a bit. So our thinking was of what this is saying is initially there was more transmission going on in Seattle than the average prediction of the model would allow for. And so to test that, one of the things that we did was to run the model for 2 weeks. And because you use a particle filter, you're updating your system state through time. We can use the initial conditions then from January 26 instead of January 12. And then from then just integrate the model forward. So by getting our initial conditions right, we actually find that our median is reasonably well matched with the data. More importantly, that the data could be described as a random realization of our model prediction. So with appropriate model initialization, so there were more cases than that one individual that we initialized, and that may well be because soon after that initial index case, there was lots of reports of COVID in nursing homes in Snohomish County. So there were probably a super spreader event that seeded really the epidemic in Washington County rather than that one individual who came back from Wuhan. One of the perhaps slightly surprising aspects of, well, I found it surprising. I'd be curious to see if anyone else does. So here are the counties that I showed you before, and underneath it, I'm showing you the splines that we fitted for each. And I had shown you previously the blue lines. Now the orangey kind of colored lines are ones where we turn off commuting. And certainly for the 3 largest populations, you take a little bit of a hit in terms of the initial baseline R0 in those populations. But there really isn't much of a difference in the splines that we're fitting for those kind of source populations. And what I interpret that to say is that once the epidemic was seeded in these large populations, probably commuting wasn't all that important. So as I mentioned, this is ongoing work, and I haven't had an opportunity to show you everything that we've done, but I will speculate about some of the implications of this. So there are some clusters in terms of the similarity of different counties in Washington state, as I showed you. There are a number of parameters like baseline reproductive number and other model parameters that are really surprisingly well identified and quite precise. So if we get this seeding of the model is sufficient to fit the model and to produce model dynamics that are consistent with the observed epidemics spread. But the initial takeoff was in the data is faster than the typical model realization, and I say that's we interpret that to be due to a super spreading event, which isn't currently implicitly represented in our model. The probability that an individual younger than 60 ends up being hospitalized is pretty well identified, and comes out to about 1%. And so we find that the commuting patterns doesn't seem to generate these kind of highly synchronized epidemics. The state is not one well-mixed population. We don't really see classic signs of a wavy type thing happening. In some of those populations right around, some of those counties right around Seattle, we find that their epidemic patterns are very similar, probably because the timing of the virus introduction was very similar, and the local transmissibility, at least when we fit them, they seem to be very similar, probably due to both kind of a mixture of social structure and also probably public policy in curtailing transmission. In terms of actual number of cases, we find that once the epidemic is initiated, we don't see any fade outs, which is interesting and important. And one of the things that it tells us is that interventions, like future interventions, non-vaccine related intervention, non-pharmaceutical interventions that are aimed at kind of breaking up or reducing the mobility network, probably won't have a multiplier effect, because it seems like mobility is not the most important thing. Once the epidemic's been introduced and has seeded the pandemic in these counties, mobility is not that big a deal. Endogenous transmission is really amplifying what's happening locally. And so we're now in the process of thinking about what this means in terms of the spread of variants and implications for immune evasion and so on. So with that, I will thank my collaborators. I will also let you know that we're very excited to have recently been awarded a Center of Excellence for Influenza Research at UGA, which for alcoholic reasons we've called CIDR. And we are looking to recruit postdocs. So if anyone's interested, or if you know of anyone who's interested, please send them my way. I'm happy to answer questions. This work's ongoing, so please be gentle.
That's great.